Hey Satterton, in this YouTube lesson we're going to be talking about the two stages in protein synthesis, which are transcription and translation. So on this slide what we're going to focus in on first is the first stage of protein synthesis, which is called transcription. And transcription, in simple terms, is RNA synthesis. So what does that mean? It's where RNA is being produced and made. The function of transcription is where a single strand of RNA is made from a specific sequence of DNA. So literally, in transcription, we're going from a specific sequence in DNA and we're making a copy of that in the form of RNA. Okay, so that's all that's going on. And just think about the word scripting. Scripting means to write. So what are we writing down? We're writing down the DNA sequence, okay, that codes for a specific protein that needs to be made. So where does this process occur? Where does transcription occur? It occurs in the nucleus. And the reason why it occurs in the nucleus is because DNA is so important, okay? It contains all the important genetic material needed to make proteins, okay? So for example, if that DNA leaves the nucleus, okay, it's increasing the chances of it being damaged. And if it gets damaged, then that means our DNA in our cells changes as well, and that's how mutations arise. So just to play it safe, transcription is going to occur in the nucleus, and that is because if DNA were to leave the nucleus, it could be damaged, okay? So literally what I have here is the DNA is kept safe in the nucleus while the RNA is sent out into the cell to direct the building of proteins. So to alleviate this process, what we're going to do is we're going to make a copy of that DNA in the form of RNA, and that RNA is then going to leave the nucleus because it's just a copy. It's okay if it gets damaged, and it's going to leave the nucleus and go to a ribosome to be translated into a protein. Okay, so here in transcription, we're going from DNA, we're looking at a specific sequence in that DNA called a gene, and that gene is going to code for a trait. We're going to make a copy of that sequence in the form of RNA, and that is transcription. So on this slide we are going to be talking about the steps of transcription and how we make a copy of a specific sequence of DNA and make it into RNA. So first things first, what we're going to see is RNA polymerase, which is an enzyme because you see it's ACE, is going to attach to DNA at a special sequence that serves as a starting signal. Okay. Notice that it's not DNA polymerase, it's RNA polymerase. So step one, RNA polymerase is going to attach to DNA, okay, at a special sequence that serves as a starting signal. The DNA strand is then going to be separated by RNA polymerase, and one strand will serve as the template strand. Okay. RNA polymerase will then add the complementary bases using the DNA template strand to create mRNA. So what you need to know right now is that RNA polymerase is going to go to a special sequence in a DNA molecule that needs to be copied. Okay, RNA polymerase is then going to unwind that DNA molecule. Okay, and then once it is unwound, the RNA polymerase is going to add the complementary base pairs, okay, using the DNA template strand to create the mRNA strand. Okay, when the mRNA strand has fully been transcribed, meaning copied, the mRNA molecule will leave the nucleus and will travel to a ribosome in the cytoplasm to be translated. Okay, so let's take a look at that. So here is just a simple diagram, okay, of transcription. Okay, and essentially what we're looking at here is you're looking at this big structure, this enzyme called RNA polymerase, and what RNA polymerase is doing is it's traveling down. Okay, first it's unwinding this DNA molecule. Okay, once this DNA molecule is unwound, okay, RNA polymerase is going to travel on down, okay, this DNA template strand, and essentially what it's going to do is it's going to add the complementary base pairs to create our RNA sequence, which is simply a copy of this DNA strand. Okay, and so if you look down here, what it's doing is, so here's our template strand that is being transcribed. You go from, so what RNA polymerase is doing, it is adding the complementary base pairs. So RNA polymerase added an A, which bonds to a T, a U, which bonds to an A. So remember, when we're creating RNA, we no longer have T, so it's going to be a U. Okay, then it's going to add a G, which bonds to C, A bonds with T, U bonds with A, C binds with G. Okay, so that's essentially what RNA polymerase is doing as it travels down the DNA template strand to make a copy of RNA. Okay, so again, here's a simple diagram of transcription. Okay, so again, here's our DNA molecule. This is our sequence. This is a gene. Okay, and this gene codes for a trait that needs to be simply transcribed into a protein. So what we're going to do is RNA polymerase is going to unwind this DNA sequence, and essentially what it's going to do is it's going to travel on down this DNA template strand, and it's going to add the complementary base pairs. Okay, so if we were the RNA polymerase, T would bond to A. Okay, A would bind to U because we don't have T anymore. C would bind to G. T would bind to A. A would bond to U. G would bond to C, and so forth if you continue all the way down. Once this mRNA sequence is fully transcribed, then it can then leave the nucleus, and this DNA molecule will close back up and it will continue its daily function. Okay, so here's just a think, pair, share if you want to go ahead and jot this down on a piece of paper and practice. Okay, but essentially what you need to do is you're given the DNA template strand. Okay, so think of this as like a DNA sequence that codes for a specific protein, and what you need to do is you need to add the complementary base pairs to create your mRNA sequence, and your mRNA sequence is a copy. Okay, and how do we get this? We get mRNA by using the enzyme RNA polymerase. Okay, so essentially if you go ahead and do this, A is going to bind with U, A binds with U, T binds with A, T binds with A, C, G, T, A, U, excuse me, U, G, C, A, U, T, A. Okay, so there is transcription. I want you to practice that a couple of times, get used to it. But really what I want you to understand is that in our mRNA sequence, we no longer find the base pair or the base T or thymine. We always are now going to be using uracil in our RNA. Okay, and remember that RNA is just a single strand. Okay, so now we're going to talk about the second stage in protein synthesis, which is called translation. Translation is the process in which we go from mRNA to proteins. So where does this process occur? It occurs in ribosomes. So remember, ribosomes are the organelle in which proteins are made. And essentially the function of translation is where pieces of RNA called codons code for specific amino acids which then make proteins. Okay, so essentially what you're going to see in translation is this mRNA sequence, okay, is going to be read 3 bases at a time, and essentially those 3 bases, okay, are going to code for a very specific amino acid. And when we continue to code this mRNA sequence 3 base pairs at a time, what we're going to start to do is we're going to start to form and bond together a whole bunch of amino acids, and those amino acids essentially are going to make up our fully functioning proteins. So in translation, a general overview that we always do and use is where we go from RNA into proteins. So here are the general steps of translation. So first what we're going to do is we're going to run through this, then we're going to talk about it in a diagram. So in the first stage of translation, the mRNA is going to leave the nucleus, and it's going to go into the cytoplasm and find a ribosome to be translated. Okay, so that is step 1. mRNA leaves the nucleus after being transcribed, and it's going to go into the cytoplasm of the cell, and it's going to go find a ribosome to be then translated. Step 2, once the mRNA finds a ribosome, it's going to enter that ribosome, and that mRNA sequence is going to be read 3 base pairs at a time. Okay, So the ribosome is going to read that mRNA sequence 3 base pairs at a time, and those 3 base pairs are called codons. And codons are groups of 3 nucleotides, better known as bases, okay, on the mRNA sequence that code for specific amino acids. So mRNA sequence enters the ribosome. The ribosome is going to read that mRNA sequence 3 base pairs at a time, and those 3 base pairs are called codons. Each codon codes for a very specific amino acid, and we'll talk about that later. Step 3. What we find then is once our codons are beginning to be translated, what we're going to start to do is we're going to need tRNA molecules or tRNA molecules that need to carry amino acids to the ribosome, okay, so that we can begin assembling a protein. Okay, so essentially what tRNA does is it is going to pick up amino acids from the cytoplasm, and it's going to bring them to the ribosome, drop them off, and then leave. So essentially tRNA are essentially transportation molecules, okay, and pick up amino acids, drop them off to begin assembling amino acids. The tRNA is going to carry an anticodon that matches the codon on the mRNA strain. Okay, so essentially how this tRNA drops off amino acids into the ribosome, it's going to contain an anticodon, which is going to bind to the codon on the mRNA sequence. Okay, and once the anticodon bonds with the codon on the mRNA sequence, it's going to drop off that amino acid. Once it drops it off, it's going to leave, and it's going to leave the amino acid there. Last but not least, each amino acid is then dropped off and a chain forms. When that chain is complete, it's going to disconnect and make a protein. So let's check that process out now. So here's a diagram of translation. Okay, and what you're looking at here is you're looking at an mRNA sequence leaving the nucleus. So this mRNA sequence was just transcribed. It's entering the cytoplasm and it's looking for a ribosome. So we're going to actually look down here at this mRNA sequence, okay, and this mRNA sequence, okay, has already entered a ribosome. So here's our ribosome. Okay, so what you're actually seeing here is you're seeing the mRNA sequence being translated in this direction to the left, okay? And remember I talked about the word codon. Here is our codon, okay? And so when this mRNA sequence enters this ribosome, okay, this ribosome is going to read this mRNA sequence 3 base pairs at a time. So here is our first codon, AUG. And that codon, okay, is going to code for this specific amino acid right here. Now, how did this amino acid get into the ribosome? Well, what happened was this amino acid was once out here, okay? And when this mRNA sequence entered this ribosome read this codon right here, AUG. Once it read that codon, it alerted this tRNA molecule right here that, hey, this codon just was read. I have the corresponding anticodon. Okay, so it's just a complementary anticodon, UAC. Now what I need to do is I need to figure out what this codon, what amino acid this codon codes for. So what it does is it uses the genetic coding chart and it finds the corresponding and the correct amino acid for this codon. It picks it up, okay, brings it to the ribosome. It bonds to this codon right here, and then once it bonds to there, this tRNA is going to leave. Okay, next, once this codon has been read and it has its corresponding amino acid, the next codon will be read. So the next codon is UUC. Okay, we're going to find the tRNA with the complementary anticodon is going to pick up the corresponding amino acid for this codon. It's going to bring it into the ribosome and it's going to drop it off. Once it drops it off, it's going to leave. Okay. Then we go to the next codon. The next codon is AAA. So this tRNA, okay, notice how it has a complementary anticodon so that this tRNA can bond perfectly to this codon right here. And what you see is that this tRNA, okay, is picking up the correct amino acid, okay, needed to make this protein. Once it has that amino acid, it's going to come here, it's going to bond to the codon, it's going to drop off that amino acid, and then once we start to see all these amino acids beginning to form, okay, in the ribosome, they're going to bond together and form a polypeptide, which is the beginning stages of a protein. Now, you're probably wondering, well, how does this codon code for this specific amino acid? And that's a great question, okay? Because if we didn't have this codon right here, this tRNA wouldn't know which amino acid to pick up. So here's how we figure it out. So this is called the genetic coding chart. The genetic coding chart is read 3 letters at a time. Each 3-letter word is a codon and specifies for a very particular amino acid. So what we're going to do is I broke down the mRNA sequence into codons, okay, from the last diagram. And so what we're going to do is we're going to look at each codon and we're going to code it using this genetic coding chart. Okay, so first things first, we're going to start with the codon AUG. So and now what we're trying to figure out is the correct amino acid for this codon. So AUG, how we figure this out is we always start in the middle. So we start with the largest letter first, so A, which is right here. Then we move to the next letter, which is U, so we move out, which is right here, and then G, which is right here, okay, which is methionine. Okay, so this codon right here codes for methionine. M-E-T. Okay, let's try the next one and then you can continue on. So step 2 is UAC, so that's our next codon. So always start in the middle, find the first letter first, so U, A, C, which is tyrosine. Okay, so T, sorry for my bad handwriting, R, Y. Okay, and last but not least, this is my last one, G, C, A, which is alanine. Okay, so if we were to continue to translate all these codons, okay, and figure out what the amino acids these codons code for, essentially what we're doing is we're assembling amino acids, and the assembly of these amino acids is then going to form a protein. Okay, and that is essentially what translation is. Okay,